I'd like to introduce our distinguished speaker, Neal Swerdlow. Neal is distinguished in many ways. He has followed a paradigm that has to do with inhibitory systems of the brain really throughout his career, and shown how it sheds light on many different disorders, especially schizophrenia, but also Huntington's, OCD, autism, and I think he'll mention something about all of those today. He's distinguished also for his productivity. He has published over 400 articles, and I was going to read the titles of each article this morning, but he asked me to keep the introduction short. I've known Neal for a number of years through his program at UCSD, and in recent years have been fortunate to collaborate with him as part of the consortium on the genetics of schizophrenia. He's a amazingly decent guy and a great leader in a collaborative venture where there are many prominent investigators. He plays a major role in keeping the collaboration on track and sane. And if you've been in many large consortium, you know they don't always stay that way. So he has both the personal characteristics and the intellectual strength to lead in many ways. So I'm honored that he's agreed to come up today, and I think we'll enjoy his talk. Thank you, Neal.
Thank you, Keith. Thank you very much. Thank you, Keith, for inviting me here. Is this too loud? Loud enough? Okay, good. All right, thanks. So to talk to you about neural circuitry and mental illness informed by models of sensorimotor gating, Keith asked me to talk about some of my newer stuff. I don't have that much newer stuff, but I've added "and therapeutics" to the title just to make it seem like I'm doing something different. But I'll be giving you some background, talking to you about disorders of impaired sensorimotor gating, how we study them in cross-species models, and then where we're going in the future. Have a lot of people to thank for my work, some great colleagues. I just want to mention Greg Light and Dave Braff, a great lab staff that I've worked with, a lot of trainees, very supportive family. Thank you, family, and my disclosures for my funding sources. So first, from a definition standpoint, what is sensorimotor gating? Sensorimotor gating is the automatic, so it's not learned, inhibition, we use the term gating interchangeably with inhibition, of a motor reflex by a weak sensory event. So sensory event inhibits a motor response, and we call that gating. The first studies of the brain circuitry for reflex inhibition were conducted by Ivan Sechnov in Russia in humans and frogs. This is his frog preparation on the bottom left. These are his drawings. You see here the picture of a suspended frog with a foot in a cup of sulfuric acid, which triggers a withdrawal reflex. And that withdrawal reflex was inhibited when Ivan put salt crystals onto an area that they called Sechnov Center, which was the thalamus. And this inhibited the reflex withdrawal response. He did this in humans in a slightly more humane preparation, but involved putting a finger in a acetic acid solution, and the withdrawal reflex from the acetic acid was inhibited by a sensory pre-stimulation. The best one was tickling the belly. And the key points that Sechnov brought up in his articles, the first one which was rejected because it was viewed as too materialistic, but then in subsequent publications, was that higher brain centers inhibit lower brain centers to provide control over primitive automatic responses. And Ivan was so bold as to say that this process applied to thoughts as well as reflexes. As he put it, a thought is the first two-thirds of a psychical reflex. He then said that to the degree that this mechanism of inhibition was sensitive to learning, it provided a biological path for psychological growth. And to the degree that this process was volitional, it provided a biological basis for free will. And then many years passed. There was a revolution, a world war, another revolution, a Great Depression. And over the next 50 years or so, this notion of automatic inhibition of reflexes by weak sensory events was rediscovered by a variety of different people. I'll just mention a few. Helen Peake, who was a postdoc at Yale, in this picture showed that you could make Yale undergraduates blink by using this gizmo to swing a wooden spoon at their face. And then you could inhibit the blink response by delivering a click to their ear. Hoffman and Fleshler used 6 Penn State rats to publish a paper in Science magazine. 6 rats in Science magazine. On the y-axis here is the amplitude of a startle reflex, and you see that the startle reflex went away if it was delivered over a background of weak pulsed noise. So the pulse 1 per second sound inhibited the startle reflex. And as an undergraduate, I did a study showing that 1 per second tail pressure caused the startle reflex, again on the y-axis, in rats to drop by about half. I had absolutely no explanation for this, knew nothing about these findings, and in fact this was counter to my predictions for my studies that I was doing. But my good fortune was that we had 3 journals in the Amherst Neuroscience Library, one of which was Experimental Neurology. That the first year that I came up with those findings, Jan Stevens published a paper showing that photic stimulation in the cat activated not only the expected portions of the visual system, but also an area in the basal forebrain called the nucleus accumbens. Now, like most people of my age and interest at that time, I knew about the nucleus accumbens from reading Jan Stevens' AGP paper on the anatomy of schizophrenia, where she called the nucleus accumbens the limbic striatal gate, along with this drawing from Wally Nauta, which showed the nucleus accumbens here, and which caused Daniel X. Friedman, a beloved member of this department for many years, to comment that no one knows where the nucleus accumbens is. True quote from him. And, but I knew where it was because it was in the Pellegrino and Cushman rat atlas. And when I went and made radio frequency lesions of the nucleus accumbens here, instead of the tail pressure, pulsating pressure, reducing startle significantly, it blunted that effect significantly. And what I'm going to tell you as a take-home message 40 years later is that there's an inhibitory window. It's activated for about 1/3 of a second after a sensory event. It's impaired in a group of brain disorders, in some cases linked to functional deficits in those disorders. It's used in animal models to predict therapeutics, and it's regulated by an identifiable forebrain circuit with a prominent role for dopamine in the nucleus accumbens. And it's regulated by the expression of certain genes within that nucleus. So that's going to be the take-home message today. But first I want to talk about the big picture. So as many of you know and have studied, our brain gets more information at any given time than we possibly know what we can do with. Right now you're getting the pressure of the chair up against your back, you're getting the taste of the coffee in your mouth, you're getting the awareness that I didn't get a cup of coffee and that could be a problem in this lecture, and you're able to inhibit automatically, without thinking about it, or gate is the term, that information so you can focus on the only relevant thing that's happening right now in your life, which is what I'm talking about. And your ability to do that, for better or worse, determines how well you're able to navigate through the world. There are a number of different neuropsychiatric disorders where the inhibition of automatic movements is profoundly impaired, and automatic inhibition of other forms of information are impaired. This is Angel, at this point a 29-year-old former defensive back with Huntington's disease. This is a disorder where you can actually see the loss of inhibition because the circuitry that's impaired, starting with the medium spiny GABAergic neurons in the caudate nucleus, ultimately connect to a motor neuron. So we can see the loss of inhibition in the form of chorioathetosis. So I use this as a visual image for medical students and residents to teach them about a loss of inhibition. But in most of our cases in psychiatry, it's not quite as visible as that. Patients with Tourette syndrome tell us they can't shut out unwanted sensations. This is a body map from Jim Leckman's work showing what we call premonitory urges or sensory tics in 100 consecutive Tourette kids. They experience this as a sensation at the skin or below, or a pressure that builds like a sneeze, or the tag on the back of your shirt. Patients with obsessive compulsive disorder can't shut out unwanted images or thoughts. A medical student with OCD told me that the image she had was of her mother. When she'd hug her, her head would fall off. The worst possible image. Schizophrenia patients tell us they can't shut things out. They told that to McGee and Chapman in their seminal paper on sensory gating in '61. They told it to Greg Light and Dave Braff in a paper they reported in 2000. So there's a group of different disorders that are characterized by, now I'll use a buzzword, transdiagnostic domain of deficient inhibition in either motor, sensory, or cognitive domains. They cross the boundaries of the DSM, but fit nicely into an RDoC framework for sensorimotor domain. I first started thinking about the neural circuitry of motor inhibition in neurophysiology class in grad school at UCSD, taught by this man, Phil Groves, former UCLA Bruin, and a person who developed in 1983 a model for the functional organization of the neostriatum and its control of voluntary movement. And his model focused on a lateral inhibitory matrix shown here of these medium spiny or spiny 1 GABAergic neurons in the caudate that served essentially as a filtering mechanism for descending input and information coming from cortex and thalamus. At the same year, Penny and Young put out a model that was based on 3 interconnecting loops which had nodes within cortex, striatum, pallidum, and thalamus, or CSPT loops. And this model was a little different. It provided an iterative way to modulate or regulate corticothalamic positive feedback, which provided a mechanism for changing ongoing movements and posture in an iterative way. And so my assignment in this course was to compare these models, and I liked this model better, but Phil was grading my paper. And so I did the only rational thing, which was I combined the 2 models. The first thing I did was moved it out of motor disorders like Parkinson's and dyskinesias and moved it up into disorders that were of more interest to me, but maintained the same 4 key nodes of limbic cortex, striatum, pallidum, and thalamus, moving it into the limbic system. And then built into the middle of it, Phil's lateral inhibitory matrix, which allowed for some predictions about how changes in different transmitter systems at different levels of the circuitry could alter the continuity of the positive feedback between the limbic thalamus and limbic cortical structures, making it either rapid switching or overly rigid in different cases. And like most models, this model was wrong. And it's been modified many times. This happens to be Jonathan Mink's version, and it's circa about 2000. It still has the same 4 key nodes of cortex, striatum, pallidum, and thalamus. There's an iterative capacity to go back and re-regulate the thalamocortical feedback. And this is the circuitry that's abnormal in these disorders that I've described. In Huntington's, the loss of the medium spiny GABAergic neurons in the head of the caudate. In Tourette syndrome, there's abnormalities in terms of striatal volume and white matter in other portions of the cortex striatal projections. With an OCD study done by Jeff Schwartz, whose office is a couple halls from here, showing hypermetabolism in the orbital frontal anterior cingulate and head of the caudate nucleus, which he later showed could be normalized with psychotherapy with CBT. I think one of the foundational findings in modern neuropsychiatry, highly influential. And then certainly within schizophrenia, there's abnormalities within frontal cortical, mesial temporal lobe, and thalamic structures. So this circuitry features heavily in a variety of disorders that are characterized by a loss of inhibition or gating. And the question is, how do we study this? How do we study the biology of these inhibitory deficits in brain disorders? And the good news is that across many different levels of brain organization, the mechanisms that get activated by one event inhibit the responsivity to a second event. And that's essentially the basis for this paired pulse inhibition paradigm that a lot of us study. You can study it in different temporal windows. Certainly refractory periods are an example of this at a very quick window. The window that I'll focus on today is this 30 to 240 millisecond window using a paradigm called pre-pulse inhibition. And so now let me talk about pre-pulse inhibition. So I'll start with the startle reflex, which is the motor reflex that gets inhibited. Analogous, if you will, to that frog reflex with the withdrawal response that Sechenov described. This is the facial and skeletal muscle component of the acoustic startle response drawn by Landis and Hunt in '39. The startling stimulus was a gun being discharged behind the subject's head. And the higher the caliber of the bullet, the bigger the startle response. We can't get IRB approval to do this kind of things anymore. But you see the facial grimace, if you will, the bowing of the back with the skeletal muscle contraction, the shortening of the torso. In the rat, it's the same reflex oriented 90 degrees differently. Again, with the shortening of the skeletal muscle frame, the back bows up, the equal and opposite force gets driven through the feet, and we quantify that, and I'll show you how we do that in a minute. Humans, we use EMG of orbicularis oculi, the big muscle around the eye that we have because we don't have a long snout to protect the eye from the poke, so got to close the eye quick. And this EMG of orbicularis oculi looks like this. So here's a schematic of this acoustic stimuli. We start with a background of about 70 decibels and superimpose a square wave pulse about 50 decibels above that. This is 0 to 250 milliseconds, and at around 60 or 65 or 70 milliseconds, in this white area, you see the EMG response to this intense, startling stimulus. And in the black area up above, you see this response to an identical stimulus, which in this case has been preceded about 100 milliseconds by a 10 decibel over background pre-pulse. And so if you visually subtract the black from the white, you have the amount of inhibition that's been generated by this little pre-pulse. And that's sensorimotor gating or pre-pulse inhibition. Conceptually, what happens is about 30 milliseconds after that first pre-pulse gets into the brain, for whatever reason, we don't understand, the brain sets up this protective period of time. And any second stimulus that arrives during that inhibitory period has a blunted response. It really has to prove itself as the brain is sort of metabolizing that first sound. Importantly, the first 50 to 60 milliseconds we can't really appreciate because we can't distinguish two stimuli that are separated by less than about 50 milliseconds, at least of this nature of stimulus. So you get inhibition even though you're not consciously aware of it, so we call that preconscious or automatic inhibition. The longer latency inhibitions, we can tell you to pay attention to the pre-pulse, and that will enhance the amount of inhibition, so we call that conscious or volitional. But importantly, this is not learned. It happens the first time. It happens in your sleep. It happens with stimuli that are too close together for you to appreciate. So this is an automatic form of inhibition up to this point about here. And it would have remained sort of a carnival show event were it not for the fact that in 1978, Dave Raff, working in Notch Calloway's group at UC San Francisco, showed that schizophrenia patients have about half as much inhibition as healthy subjects. On the y-axis here is the amount of pre-pulse inhibition, and here with short, medium, and longer interval pre-pulses, with pre-pulses that are sound, but the startle stimulus is an air puff, so cross-modal. No matter how you look at it, schizophrenia patients had about half as much inhibition as they should in these paradigms. This finding's been replicated by a lot of groups, and there are meta-analyses now showing these effects. We've studied many different features. I won't go into all of them. I'll mention sex, medication, and smoking as three variables that we find consistently regulate the amount of inhibition. These are all schizophrenia patients. This is from a study in 2006. Men are more inhibited than women. People taking atypical antipsychotics generally have more inhibition than patients taking no atypicals or no medications at all. And in general, although there are some differences in these reports, smokers have more inhibition than non-smokers. The amount of inhibition grossly correlates with global function, here shown, and with a variety of different neurocognitive measures. And it differs a little bit across laboratories, but the general notion is the more inhibition you have in this reflex, the more your forebrain is working right. That's sort of the blunt way of describing it. We then reported that this amount of inhibition was deficient not just in schizophrenia, but in these disorders that I've described to you. And these are just examples of it. Huntington's patients are profoundly impaired in pre-pulse inhibition. OCD patients are impaired, but not to the same extent, and at some levels have normal levels, but they do show deficits. Both of those findings have been replicated by many groups. I just mentioned a few here. The first report in Tourette's was actually done by Javi Castellanos working in Judy Rappaport's group. We've replicated it, showing it was not modality specific with both acoustic and tactile deficits. And there's other neurodevelopmental disorders where PPI deficits have been now reported, and many other disorders where it appears to be normal and intact. What has made it very attractive to study in the laboratory is that it's present across mammalian species and can be studied across species relatively easily. So we can study it in mice, rats, using whole body startle. You can use single muscle EMG shown here. studied marmosets, macaques, kids. This was my daughter when she was about 11. There's really impressive cross-species similarities in the parametric features of pre-pulse inhibition. This just happens to be one amount of pre-pulse inhibition on the y-axis, the intensity of the pre-pulse over background, showing very parallel curves in humans and rats. Same software, same hardware, really, right up to the electrode across species. And this allowed us to use a variety of animal models of pre-pulse inhibition. This happens to be one for predicting antipsychotic potency. We showed that a low dose of a dopamine agonist takes pre-pulse inhibition from this level in a albino Sprague Dawley rat down to almost nothing, but then you could restore pre-pulse inhibition in a dose-dependent manner with either typical antipsychotics like haloperidol or atypicals like clozapine. And if you look at the ED50 for these drugs in this paradigm versus their relative clinical potency, you see it's basically a straight line. And this has allowed drug companies to use this to screen rapidly antipsychotics. And unfortunately, it's produced a lot of antipsychotics that more or less do the same thing. And so that's the downside of having a model that has this level of predictive value. We've used it, and others have used it, in models with construct validity. So one construct for the pathophysiology and schizophrenia has to do with some early developmental or even in utero mesial temporal lobe abnormalities. Barbara Lipska and Danny Weinberger developed a model of neonatal ventrohippocampal lesions, day 7 rat pups that then grew up to exhibit some interesting behavioral phenotypes, one of which was, we showed in 1995, deficits in pre-pulse inhibition that developed post-puberty in these rats. So at day 7, if you take out their ventral hippocampus, which is shown as a lesion here, they grow up, and post-puberty, their pre-pulse inhibition essentially goes away. That was shown here by a French group who replicated this effect. Here's a healthy rat that grew up with a healthy ventral hippocampus. If you take out their ventral hippocampus as day 7, you see they lose about half of their pre-pulse inhibition, and then you can restore that in a dose-dependent manner with clozapine, shown here. This is the human analog done by Veena Kumari's group in London, compared to healthy subjects. Schizophrenia patients on typical antipsychotics have about half as much inhibition, and you can normalize it in them with clozapine. Other groups have shown that this form of inhibition appears to be under tight genetic control, although that doesn't mean it's simple genetic control. I think one of the more interesting reports was done by Tom Insel when he was still at Emory. And Tom's group took 2 strains of mouse, a black 6 and a Balb C. They had a variety of different behavioral phenotypes, but if you focus on PPI, the black 6 were high pre-pulse inhibition, the Balb Cs were low pre-pulse inhibition. And if you took an embryo from a black 6, transplanted it into a Balb C uterus, and had it reared by a Balb C mom, they still grew up to have a black 6 PPI phenotype. So this is a good example of nature beating out nurture. We already knew that even one gene could profoundly impact pre-pulse inhibition from our work with Huntington's, which is just the same data I showed before, just replotted here. Rebecca Carter's group showed a couple years later that you could actually create this de novo in a mouse that was transgenic for the Huntington's gene, which by about 12 and a half weeks of age completely lost their pre-pulse inhibition. I spent time studying the heritability not of pre-pulse inhibition per se, but of the sensitivity of pre-pulse inhibition to disruption by a variety of different drugs, if you will, the resilience of pre-pulse inhibition. This is one example of the kind of studies that we did this with. This albino Sprague and hooded Long Evans animals, both from Harlan Laboratories, had really substantially different phenotypes in terms of their sensitivity to the disruption of pre-pulse inhibition. If you take the albino here and the hooded animal here, you see their overall level of inhibition doesn't really differ. But given a low dose of a dopamine agonist, these albino Spragues completely lost their inhibition, whereas the Long Evans hooded animals maintain a fair amount of inhibition. Dopamine doesn't seem to really disrupt their gating that much. You could cross them to make an F1. We call it a long dolly. And the long dollies had an intermediate amount of dopamine agonist sensitivity. And if you back cross the N2, you see the 50-50 Mendelian split in terms of coat color and roughly intermediate phenotype in terms of dopamine agonist sensitivity. And so within really 2 or 3 generations, you could profoundly change dopamine agonist sensitivity in these animals. I'll talk a little bit more about that later. What I've shown you in these animals up till now was at around the 100 millisecond level, if you look at this, the gap between the pre-pulse and the pulse. And this is a replotting of those data showing that the Long Evans animals, their PPI drops just a little bit with a dopamine agonist, whereas the Sprague animals, the inhibition drops profoundly. But the interesting phenotype actually happened if you look at the full window, and so remove the rectangles there, and you see that once you get past about 30 milliseconds, moving backwards in this gating window, dopamine agonists actually enhance gating in this strain of animal. And so dopamine is doing 2 very different things in terms of the temporal window of inhibition, and presumably in terms of the type of information that would be processed at those different time points. Certainly very different than the albino Sprague. And this has allowed us to use this type of a strain difference to understand brain circuitry, which is where I'm going to be focusing most of the rest of the talk on. Fortunately, Mike Davis then at Yale had defined much of the primary startle circuit. And what we did was basically use the readout from here, kind of like a telescope looking through a Pontine portal up into the forebrain, and we would make lesions or do drug injections or stimulate different parts of the forebrain, and the signal we would get in terms of this change in the startle reflex would be basically coming out of the pons, and we could make manipulations and understand the anatomy that way. I'll focus, so we use a lot of different techniques. This is about 20 years, different experiments. And I actually learned to do this stuff at the Brain Research Institute, 78 and 79, working with Larry Kruger. I don't know if anyone in here knows Larry. But he taught me how to do neuroanatomy and stereotaxic surgery. And we use a variety of techniques, lesions, microdialysis, just drug injections. These are neonatal ventriculohippocampal lesions. Axonal tracing techniques, silver staining we've used to this, to try to connect from prefrontal cortex down into the Pontine circuitry that regulates pre-pulse inhibition. And I'll just focus on 2 parts of this circuitry. I'll start here. This is the projection from the nucleus accumbens into the ventral pallidum. This is that GABAergic medium spiny projection that I've talked about already in terms of the dorsal circuitry for Huntington's disease. This is where presumably some of the dopamine receptors are that we've been stimulating to disrupt pre-pulse inhibition. And what you can see from putting a dialysis wick into the ventral pallidum is that if we use increasing doses of a dopamine agonist, that there's a dose-dependent reduction in the amount of GABA efflux in the ventral pallidum. So if we stimulate the dopamine receptor there, it reduces GABA release 1 synapse downstream. And that's really what's happening. You can see a replication of that here in these Sprague animals. This is where apomorphine was infused. And this is a placebo. This is the apomorphine effect, and it cuts GABA efflux 1 synapse downstream down by 20–30%. That doesn't happen in these hooded Long Evans animals. Remember, these are the ones for whom apomorphine doesn't disrupt gating, or not very much, and for them, apomorphine also didn't disrupt GABA efflux 1 synapse downstream. So something has changed between these 2 strains, between the dopamine receptor and the release of GABA downstream. And we followed that through different signaling pathways using the paradigm where we would study the parental strains and backcross and make an F1. And we found across many different levels of signaling pathways within this accumbens ventral pallidum projection that the same pattern that we detected with pre-pulse inhibition was reproduced within the signaling pathways. We work with Ron Hammer, another former Bruin, to do the work at the level of GTPS binding, and that's very early in the signaling pathway. Later in the pathway with Krebs phosphorylation, you can see here compared to a vehicle injection, this is the inhibition of Krebs phosphorylation with apomorphine, and the amount to which apomorphine disrupted pre-pulse inhibition corresponded relatively nicely to the amount to which it inhibited Krebs phosphorylation, and the strain differences in these effects were reproduced. So here you have sort of a complex behavior, and a very simple sort of molecular phenomenon that's happening inside that GABAergic neuron and very close parallels. We were dealing with transcription factors. We looked at gene expression differences across these strains. So these are Long-Evans animals, heat maps with Sprague-Dawling animals. And we looked at expression of 100 different genes that were involved either in dopamine neurotransmission or implicated in schizophrenia, and found some fairly profound differences in the accumbens. I'll just point out a couple. A catechol methyltransferase you see here in the Long-Evans animals, substantially more than in the Sprague's at a level of 4.8 times 10 to the minus 17th. So these are big differences, and it allowed us to sort of develop some models for what's going on at the molecular level within this circuitry. But the bottom line is that something really profound has changed within this one neuron that accounts substantially, if not completely, for the differences in dopamine agonist sensitivity. I'll broaden this circuitry a little bit to include medial prefrontal cortex and some of the subcortical dopamine circuitry. We've known that medial prefrontal cortex regulates prepulse inhibition since the 90s. Michael Koch's work in Germany showed that if you just deplete dopamine from the medial prefrontal cortex, protecting norepinephrine, you see a drop in prepulse inhibition in rats. You can then restore that gating with haloperidol. A couple of years later, we showed that that effect was likely due to selective inhibition of D1 receptors because we infused the selective D1 antagonist, SCH23390, and could reproduce this drop in prepulse inhibition just by blocking prefrontal D1 receptors. The sort of cartoon model of how we thought about this comes really from Chris Pycock's work in the 70s, showing this essentially reciprocal cortical-subcortical dopamine regulation to the degree that it's real, showing that either if we depleted dopamine or blocked the D1 receptors at a cortical level, that there was a subcortical reciprocal upregulation or increase in dopamine tone that was then responsible for a loss of prepulse inhibition. We could study this a little bit more in depth using a variety of different antipsychotics. Here, this Long-Evans animal, given a dopamine releaser, amphetamine, you see a very little reduction in prepulse inhibition with amphetamine at the longer intervals, the increase in gating at the shorter intervals. But importantly, if we block D2 receptors with antipsychotics like haloperidol and even the atypical quetiapine, amphetamine actually now increases gating. And it does it there, and it does it there as well. And so the model here would be that we're blocking with antipsychotics the subcortical D2 tone, driving dopamine release with amphetamine. It's predominantly hitting those cortical D1 receptors, which is enhancing gating. And we can flip the equation by giving that D1 antagonist, SCH23390. In this case, we're blocking here. That drives the subcortical tone, which reduces prepulse inhibition, and that's what you see here now. Amphetamine, rather than enhancing gating, actually suppresses gating. This is of relevance because we can do this in humans. And this is a study where we have schizophrenia patients taking antipsychotics. Given placebo, they have a deficit in prepulse inhibition. But now that we've blocked the subcortical D2 tone, we give them amphetamine and it actually enhances gating. So the model cartoon explanation here, again, is the same as I just showed you. Blocking subcortical D2 activation, amphetamine releasing dopamine, the prefrontal tone predominates, and we see an increase in PPI. There were two interesting correlates that we detected. One was with chlorpromazine equivalence. The more antipsychotics on board, the bigger the increase with amphetamine. And the other was with amphetamine-enhanced happiness. So we have every hour our subjects make a mark on a line, a visual analog scale for their happy as well as alert, dizzy, lots of other feeling states. And the ability of amphetamine to increase gating actually was inversely related to its positive hedonic effects. And we don't have a real answer for these two, but a simple parsimonious explanation is that the less D2 blockade here and the more amphetamine activation of the subcortical dopamine tone, it prevents amphetamine from enhancing gating and it produces the positive hedonic effects. So again, this is still working with that sort of reciprocal PICOC cortical subcortical dopamine tone model. But it's clearly the case that amphetamine is actually producing a bigger effect the more antipsychotics that are on board. So the take-home message, there's an inhibitory window. It's activated for about 1/3 of a second after a sensory event. It's impaired in a group of brain disorders. In some cases, it's linked to functional deficits. It's used in animal models to predict therapeutics. It's regulated by an identifiable forebrain circuitry with a prominent role for dopamine in the nucleus accumbens, and it's associated with the expression of certain genes within that nucleus. So what's next? This is one of my favorite quotes from Men in Black. This is Tommy Lee Jones telling Will Smith, who had just seen Aliens for the first time, imagine what you'll know tomorrow. And so what will we know tomorrow? I had no clue that deep brain stimulation would be a thing when I started doing these studies, but a lot of people are now using PPI to predict sites within the brain for therapeutic interventions with DBS. Kirsten Schwab's group and others have done this. This is her data of 2 strains of rats that have been selectively bred to have low or high levels of prepulse inhibition. You can do this in about 3 generations. It really changes that quickly. And if you stimulate thalamic structures, there's a current-dependent increase in gating in the poor gators. This is another study done by a different group where PPI was disrupted by giving in utero immune challenge. And you can see that compared to a control group, the in utero challenged group had about half as much gating, and you could restore gating in those animals with medial prefrontal cortex stimulation. And these are 2 of many examples that have been mapped out here showing cortico-striatopalatal thalamic circuitry where you can stick an electrode and stimulate and normalize gating that's been disrupted by a whole variety of different things. Ketamine, immune challenge, selective breeding for high or low, apomorphine, and others. So there's essentially, there's many ways to disrupt gating, and we know that because there's many disorders where there's deficient gating. And there's different stimulation sites within the CSPT circuitry that appear to correspond to ways to normalize the gating in those conditions. And this is being done at the clinical level in OCD, where DBS in the nucleus accumbens has been shown to have therapeutic effects. Control subjects, OCD patients pre-op, OCD patients with the nucleus accumbens stimulator turned on and turned off. So prepulse inhibition is deficient in the patients, normalized by DBS in the nucleus accumbens, and the deficit returns when you turn the stimulator off. Lest I leave you the notion that it's only DBS that's useful as a PPI for predicting DBS therapeutics. This is a study done by Veena Kumari's group showing that the amount of symptomatic improvement with CBT in schizophrenia patients correlates rather strongly with the amount of prepulse inhibition at these longer sort of conscious intervals. So something about the way the brain is processing that information is associated with benefiting more from CBT. And the last example at a therapeutic level I'll mention, so lest I leave you with the notion that this amphetamine-enhanced gating is only good for studying PPI, we asked the question whether prefrontal dopamine, activating prefrontal dopamine tone using this approach might enhance learning in schizophrenia patients. We used the Sound Sweeps component of Posit Science, a computerized cognitive remediation program that Sofia Vinogradov and many have used so successfully, where subjects hear whip boop, and they have to press an arrow, depending on which direction the sweep is in. As they get it correct, the sweep amplitude reduces, the gap closes, allows you to calculate an auditory processing speed. We get a pre-training level and an hour of learning, and then a post-training level, and you can then detect the amount of gain or learning that happens at an automatic implicit level with this paradigm. And this learning is, in schizophrenia patients, is increased in an inverted U function by amphetamine. And so this is a study that we have ongoing now. We've done single dose and dose response functions. It's not state dependent in that this is the carry forward learning a week later, and the gains that were made under amphetamine from the previous week are retained at least for a week. So lastly, we're trying to use pre-pulse inhibition as a biomarker for precision medicine studies. You all know the story here. If we use a clinical phenotype to develop a drug and apply an intervention to a heterogeneous population, there may be some individuals there who are highly sensitive, but that overall that gives you a failed study. Whereas if you can identify those individuals ahead of time using a biomarker that detects their sensitivity and just intervene with those, you have a treatment success. And we're asking whether drug sensitivity of pre-pulse inhibition and other related measures of auditory processing can be those biomarkers. And what you see, each dot here is a person, and it's the magnitude of their drug response in terms of pre-pulse inhibition and other measures of early auditory information processing. And if they're above this line, the drug is enhancing pre-pulse inhibition in those individuals. This drug happens to be memantine. And we call those people drug sensitive, and now we're asking whether that sensitivity with a single drug challenge ultimately predicts their clinical response, and that's a study that's also ongoing now. So I've told you about the neurocircuitry and some about therapeutics of mental illness informed by models of sensorimotor gating, the background, those disorders, how we study them, and what's next. And I'd like to stop now and answer questions.
So let me start.
Okay.
You showed very convincingly that amongst the antipsychotics, the pre-pulse inhibition is modified by the dopaminergic pathway. With the newer antipsychotics and oftentimes serotonergic pathways being, is there any effect from other pathways amongst the
Yes.
antipsychotics?
So I only focused on dopamine today, but Mark Geyer and his group and many others have shown serotonergic agonists of various kinds will disrupt pre-pulse inhibition. You can reverse that with serotonin antagonists. There are noradrenergic regulators, certainly NMDA function. Interestingly, when they try to cross systems, you see some interesting things. So phencyclidine profoundly disrupts pre-pulse inhibition, and we've shown it in rats. Many other people have shown it in rats and mice. Dan Javis shown it in macaques. And in each of those cases, you can normalize gating, but not with typical antipsychotics, only with the atypical antipsychotics. And clozapine appears to be particularly good at that. And so you can certainly work within the sort of the receptor tautology, which is that you give the agonist and then you give that produces the behavioral change, and we give the antagonist for the same receptor. That stuff's pretty boring, but probably accounts for how some of these newer antipsychotics are working. But the exciting stuff is when you're able to cross the systems. And so you can certainly do that with glutamate and dopamine, and probably some of the others.
Right.
Really nice talk, Neal. I was really impressed with the poly-IC data, and it really raises a question about whether other inflammatory signals will produce that downregulation of PPI responses. But really where I'm going with this question is this notion that there's a lot of variability in schizophrenia prognosis and progression. And it appears that people that have schizophrenia that have a higher inflammatory component are more likely to show a worse prognosis and progression. So I'm wondering to what extent you've actually stratified or looked at the variability in inflammatory levels in your PPI responses in schizophrenia patients.
Yeah, so I haven't done that inflammatory work. Susan Powell is doing some of that in San Diego, and this group, that the Client's data, they're not at UCSD. So I don't want to speak to the details of it other than to say that a number of groups have shown that in utero inflammatory activators, for lack of a better term, the animals will grow up to have PPI deficits. And I don't really understand the mechanisms for that particularly well. We know a lot of in utero insults and early developmental insults in rats will cause them to grow up to have PPI deficits. Certainly isolation rearing and other early stressors will produce that effect. And I agree with you that certainly the heterogeneity in schizophrenia is probably due to not just differences in the inflammatory systems, but in lots of other mechanisms that all converge onto some early developmental either neuronal migratory or developmental effect within mesial temporal lobe that then can later manifest itself, both with PPI deficits and with psychosis.
Great talk. Could you imagine that we could identify people with schizophrenia who would, based on inhibition using it as a biomarker, who would benefit from having amphetamine added to their antipsychotic? And is that something that one would do chronically?
So let me answer this second question first. We have no experience giving amphetamine chronically. Henry Nasrallah has done that, and others have done that. As long as people are taking their antipsychotics, critical caveat, there appears to be some gains, at least in terms of negative symptom relief. And that's on a daily regimen. What we've done is we've added it just on the days when people are doing the cognitive remediation paradigm. And so they're taking a very, very low dose, roughly, I don't know, Jim, 5 milligrams of amphetamine. Would you start a 6-year-old on something like that? In that range. So, and they're only getting it a couple times a week when they're doing it. So would the amount of pre-pulse inhibition predict whether we should give someone amphetamine and expect benefits? You could probably do that. I think a smarter way to do it is just to look at their matrix attention vigilance score. And my guess is that will tell you who are the people who need the amphetamine, because my guess is that what's causing the enhanced learning with amphetamine in the schizophrenia patients is it's probably improving their attention and vigilance for this paradigm. And we've seen that is one of the key determining factors to how well one learns this sound sweeps paradigm.
Hi, I noticed you had a list of conditions in which this was impaired, and a list of conditions, one slide that showed, depression, it wasn't impaired. I was just wondering where disorders like anxiety disorders or PTSD, which sort of group they fell into.
Great question. The answer is there are studies showing panic disorder and PTSD patients having deficits in pre-pulse inhibition. They haven't been as widely reproduced, maybe because of the heterogeneity in those populations or other reasons or medication effects. You can't really hang your hat on them yet. There are other disorders like attention deficit disorder where it appears that the PPI deficits are there in a paradigm where you tell people to pay attention to the pre-pulse. You don't have to tell them that. You can give sort of a warning the pre-pulse is about to happen. You use the stimulus configurations. We call it a peak on pedestal. So there's a pedestal of increased sound that warns them they're going to get a pre-pulse. And then that is deficient in ADHD. But the automatic inhibition where there's no instructional component appears to be intact. We think in Alzheimer's it's intact. Although in Alzheimer's with psychotic symptoms, there may be deficits, and people are studying that now. Bipolar disorders, findings have been mixed. And in manic phase, bipolar patients probably have PPI deficits. In various addiction groups, they appear to be relatively intact. That's the long answer. The short answer is there are people who have reported PPI deficits in both panic disorder and PTSD. Let me also just mention there's one, perhaps more surprising in PTSD is there's a number of studies that show that startle reflex is intact in PTSD, even though subjectively, clearly, that's reported as being elevated.
Neal, a great talk. Impressive body of work. Is PPI also potentially a marker of risk for psychosis before clinical symptoms are evident?
I don't know. I can tell you that in our COGS studies, you know, the amount of PPI deficit in asymptomatic first-degree relatives is probably, they probably have some deficit, but it's really not particularly robust. Christy Caden has seen it and reported it. The COGS group in first-degree relatives didn't see much. In the prodromal literature, there's mixed findings. So I don't know yet whether this is going to be helpful in identifying people who are going to go on to develop schizophrenia.
Might you be able to manipulate the test in the way you described with perhaps low-dose dopamine agonist manipulation to enhance its sensitivity for that purpose?
Yeah, and there's even easier ways by manipulating the stimulus parameters, which you can, one of the great things about this I didn't get a chance to talk about, if you're a behavioral person, you love behaviors where you can tweak stimulus parameters and conditions and selectively activate different parts of the behavior. There may be ways of tuning up the sensitivity of the paradigm that could be more sensitive for detecting milder deficits. I think there's, at a population level, I'm not going to say that the basal levels of PPI are necessarily going to be strong risk predictors. There just may be too much variability for it. If only for the fact that a lot of people who show up at a prodromal clinic go on to develop major depression, bipolar disorder, many other conditions, some of which may have PPI deficits, others which don't. So I'm not convinced of that yet. Once you then start manipulate by putting in stimulators or giving drugs, I agree that may tell us more about risk.
Thanks, Neal. Great talk. Within schizophrenia patients, what about relationships between PPI and negative symptoms, positive symptoms, specifically with the positive symptoms, hallucinations, and then just negative symptoms in general?
Yeah, so there's a variety of different reports on this one. I'm not going to try to convince you one's right or wrong. In general, I'll say the people who are most functionally impaired have the worst gating. It tends maybe to go more with, you know, more positive symptoms. Certainly that's how people viewed it as a model. I think that's an oversell. And I don't necessarily think that you can distinguish positive versus negative symptom complexes that way. Certainly when we give the amphetamine, the people who show the more positive hedonic effects are the ones who show the least gating improvements with the amphetamine. And we think that's because we're driving subcortical tone. But I wouldn't use this as a way of separating out different forms of the schizophrenia.
With the two kinds of rats you use, the Swerdlow and the long Evans,
Long Evans, yeah.
Would either one of those be more representative of humans, do you think? And then is there any role for GABA modulation, as that was one of the key factors?
Yes, I'll say maybe and yes. So first of all, I just showed 2 strains. We probably studied a dozen. They all have some similarities and there's differences, even amongst the outbreds. Once you get into the inbreds, you can dial different drug sensitivities and levels of PPI in fairly well. So I've just picked 2 out of a hat here. Not really, I picked these for reasons I wanted to be able to follow the phenotype of the coat color. It's an easy way to track things. And actually the body surface area of the pigmentation, it was a strong predictor of the apomorphine sensitivity in this. There's a whole other story behind that. But if you just look at dopamine agonist sensitivity, and I showed one slide in there where we used amphetamine challenge in the Long Evans animals and amphetamine challenge in the humans. And the Long Evans animals give a human-like amphetamine phenotype. Very low, small amount of disruption at the short intervals, a big amount of gain, a small amount of disruption at long intervals, a big amount of gain at the short intervals. And we saw that both with amphetamine in the Long Evans animals and with amphetamine in healthy human subjects. GABA is all throughout this circuitry. I mean, GABA is everywhere in the nervous system. It's not terribly surprising, but I mean, If you put picritoxin into the ventral pallidum, PPI goes away. If you put mucimol in the pedunculopontine nucleus, PPI goes away. And certainly the changes in GABA tone driven by the dopamine receptor stimulation, we think are what accounts for the strain differences. And I showed that in the dialysis data. So, and those are just a couple of examples. But throughout this whole CSPT circuitry, GABA plays a prominent role, primarily in the subcortical components. The corticothalamic feedback has primarily glutamate as a driving force. But yes, so in terms of systemic GABAergic drugs, interestingly, because it's everywhere, there's probably a lot of effects that cancel each other out, and you give it systemically, and it's really hard to make sense of it. But once you can start going fusing it point to point, the data is pretty clear.
So let us thank Neal again for our wonderful
Thanks, everybody.